Scientists have identified a breakdown in cholesterol transport to neurons in Alzheimer's disease, a defect linked to the high-risk APOE4 variant. Researchers from the St. Pau Research Institute, St. Pau, the Clinical Biochemistry Service at Hospital de St. Pau, and the Spanish Biomedical Research Network in Diabetes and Associated Metabolic Diseases, CIBERDEN, have uncovered an important disruption in how cholesterol reaches the brain in people with Alzheimer's disease. Their work shows that lipoproteins in the cerebrospinal fluid of affected patients are less effective at supplying cholesterol to neurons, a problem that may be connected to the APOE4 genetic variant, one of the strongest known risk factors for Alzheimer's. The study appears in the Journal of Lipid Research. According to Dr. Mireia Tondo, researcher in the Lipid Disorder Pathophysiology Group at St. Pau and lead author of the project, we've long known that people with the APOE4 variant, especially in the homozygous form, have a much higher risk of developing Alzheimer's. But until now, the reasons for this weren't well understood. Our study suggests that one contributing factor may be that neurons, in the presence of this variant, have a harder time absorbing cholesterol delivered through the cerebrospinal fluid. Cholesterol, essential for neuronal viability. Cholesterol is a vital molecule for proper neuronal function. It plays a key role in membrane formation, synaptic transmission, and myelin production. Unlike other organs, the brain does not receive cholesterol from the bloodstream due to the protective blood-brain barrier. All the cholesterol the brain needs is produced locally, explains Dr. Tondo, and it's stored in specific lipoprotein particles that transport it from glial cells to neurons. If this process fails, the neuron may not receive the structural and functional resources it needs. The research team analyzed cerebrospinal fluid samples from 10 patients with Alzheimer's disease and 10 individuals without the disease, all part of the St. Pau Initiative on Neurodegeneration SPIN cohort. They evaluated 2 stages of cerebral lipid transport. First, the ability of astrocytes to release cholesterol into the cerebrospinal fluid, and second, the ability of neurons to absorb this cholesterol. The results showed that astrocyte cholesterol release was similar in all participants, but neuronal uptake was clearly impaired in patients with Alzheimer's. Based on these findings, the team sought to determine whether this defect was related to genetics. Most of the patients in our sample were heterozygous for the APOE4 variant, and we observed lower cholesterol uptake in them. So we decided to go further and create recombinant lipoprotein nanoparticles, identical except for containing either APOE3 or APOE4, Dr. Tondo explains. When we tested them in cultured neurons, those containing APOE4 delivered cholesterol much less efficiently. This led us to believe that this variant may directly contribute to the dysfunction we observed.